Hello, welcome. I'm Cameron Davidson-Pilon. Welcome to my workshop. Today I'm going to present a really cute little problem for you. Came around last year and the traditional kind of solution was it hit all the interesting uncertainties, so I wanted to propose a Bayesian solution to it that I think is more interesting, and it's a nice little kind of toy problem and it's easy to extend. Okay, so the problem is I might have a culture, a liquid culture. Oh, and I'll explain all the biology as we go. There's not much, but okay. So I have a liquid culture, something like this gross thing. This is yeast, like highly concentrated yeast, and I want to know how many cells per milliliter there are, like the cell density, essentially. That's useful to know because I might want to pitch this in a beer, and I don't want to what's called overpitch or underpitch. I want to know how many cells are going into that beer so I can maybe scale the volume appropriately. I might also, I'm not talking about yeast, I could be looking at a blood culture and I want to count the number of red blood cells per milliliter. That's useful. I could be looking at algae cultures or sperm cultures. You can count sperm. Basically, what I want to do is count the number of cells per milliliter. Really useful to know. So how do I do it? So what we're going to do is we're going to take a sample of this, and we're going to use that microscope back there and something called a hemocytometer. Hemo, blood, cyto, cell, meter, count. It's this, and we'll look at it under the microscope in a minute. So we plate a small little volume on here, and there's like grid lines. We'll see that under the microscope, and we use that to manually count the number of cells. So that's really useful. And the first problem we're going to see, though, is that this yeast culture, it's too dense. There's too many cells per milliliter. If I just took this and looked at it under the microscope, it would be just millions of cells, and I can't count millions. That's just not possible. So the simple solution is a dilution. So we'll take a small portion of this and we'll start diluting it into some reasonable density and kind of work our way back up to the original density at the end. Okay, so to dilute, again, like I said, we take a small sample of this, we'll take a milliliter, and we're going to add it to, this is 9 mils of water. So it's a 1:9 ratio. And we're going to switch this around, and we're going to do the same thing to this next one. So take a milliliter of this, add it to this one, switch it around, and then do it again to this one, milliliter of this, add it to this, switch it around. These are all 9 mils approximately. And then at the end, we'll have this final dilution. It'll be much less diluted, 1,000 times. And then we'll put that on the hemocytometer, this little glass thing, and look at that under the microscope. So I'm going to do that. I'm going to record it. I might speed it up if it's too boring, and we'll look at the microscope next. Okay, cool. I'm not wearing gloves. It's not a big deal. Maybe I should be, but oh well. Okay, so take a milliliter of this. By the way, I'm using these, just crappy plastic pipettes here. They're accurate. We'll find out, but just using these for now. And we added a milliliter to this. And I like to kind of like pipette up and down to kind of get any residual yeast. Okay. Great. Okay, just give it a quick shake and do the same thing. So take a mill. Add it to the next dilution. Oops. Maybe we'll put some nice music over this. Okay, shake it. Okay, last one. Mil. That's 9 mils. Let's give it a quick shake. Okay, so we can kind of see, let's cap these again. We can see the progression of dilutions. You can see that. So we're getting, obviously, this is not a surprise, more diluted. Okay, so now I'm going to take a very small sample from the final dilution and add it to this plate. This plate, it's calibrated such that there's a known volume on here, and any excess volume kind of falls off the side. So there's a known volume on here, and that's useful to know for our later calculation. Okay, let me just do that real quick. You don't need to see this. It's as boring as it sounds. I'm just taking a very small sample and putting it under a glass plate. So it's like, when I say small, it's like 0.00001 milliliter, that order of magnitude approximately. So I just literally need a drop less. Okay, that's done. Okay, let's look at the microscope, and yeah, we'll do that in a second. So I'm going to switch over to my iPhone, which is plugged into my microscope using just like an optical piece, and we'll be able to view what's happening under the microscope on that hemocytometer. So these are the grid lines. We're gonna zoom in more to really see what's going on. Cool. Okay. So I hope you can get a good resolution on that. Yeah, that's pretty good. Okay, so let me actually zoom out one more time so we can kind of see the entire hemocytometer. So you can see that there's a bunch of grid lines. You'll note that there's 25 larger squares. So there's many small squares, but 25 larger squares. If you can't see it, take my word for it. So the 25 larger squares, we're going to choose 5 of them, usually the corners in the center square, and we're going to count the number of yeast in those squares. So zooming in once again, look at these yeast. That's what they look like, folks. That's what you drink. And so we're going to start on the top left. Okay, cool. Now there's a certain protocol for counting yeast like this. It involves, like, do you count on the edge? Which edges do you count? I'm going to offline count. It's really boring counting these. I'm going to offline count them, and then I'm going to come back to this video and tell you what I counted. But yeah, I mean, if you want to take a picture and count it yourself, but this is the idea. This is what you do. You're literally counting these little buds. One thing I kind of wanted to show off too was just, you can, this iPhone attachment is so cool. I can zoom in really close to the yeast using just, like, you know, the zoom function on the iPhone. And so yeah, you can get a pretty cool view of the yeast. Not bad at all. So I just finished counting. I counted 95 in the 5 squares. So let's get on with the analysis now. So I'm gonna share a deck. Hopefully you can see that. I'm assuming you can. Okay. So this is kind of the formula that has been historically been used. It's based on just kind of looking at the ratios and kind of scaling up the dilutions. Cells counted, or cells per milliliter, is equal to cells counted times or divided by the number of squares counted. We counted 5 times the dilution factor divided by that small volume on the hemocytometer. That's where that number is. Okay, so let's put in our numbers. So I counted 95. I counted a total of 5 of the squares. Our dilution was 1,000 times, and we divided by this hemocytometer volume, and we get a number that's 4.75 billion cells per milliliter, which is about what the yeast manufacturer advertises as well. So it's accurate in that sense. It's in the right order of magnitude at least. You can also see now why if I didn't dilute, even like a quarter of a milliliter or a tenth of a milliliter is just way too many cells. Kind of like makes you appreciate how many cells are in this original vial as well. But let's think more about this formula. Okay, so let's look at our process. Well, I first created these dilution vials. I did that using my standard volumetric flask or vial. How accurate is it? Well, it's a little bit accurate. Like I eyeballed 9 milliliters. I assume the glassware is sort of accurate. I also, if you noticed, I mentioned it, I pipetted using my somewhat reliable pipette, this plastic pipette. You can see it on that screen there. You know, who knows what the quality of these are. I could have, same with when I measured out the 9 milliliters of water, I could have used a very expensive pipette, and we'll get to that later. But this is what I have on hand, and that's kind of what I've been using. So there's already a bit of uncertainty in this dilution factor that we haven't really incorporated. And it's really important too, this dilution factor, it's layered, it's hierarchical. So an error up here propagates quite large as we make that dilution further and further, or make more and more dilutions. The next source of uncertainty is actually in the hemocytometer. So this is a picture of it. They give an estimate of the volume with a standard error there, which I assume is like it's kind of standard error that they've calculated somehow. This isn't a super high quality hemocytometer. It cost me like $10, I think. So there's lots of uncertainty in that too. So we have this additional source of uncertainty in our calculation. And again, it's because we're divided by such a small number, it could really, a small error there really could magnify our final estimate. And finally, this is a picture of drawn picture of my hemocytometer with little yeast cells on it. There's 9 squares here, but remember, we're only counting a fraction of them. So I only count cells that exist in the outer squares and the middle square. Could be by chance that a lot of the yeast cells end up outside my visible or my counting region. That's just could be by chance. So there's uncertainty there too. And so we want to model all of these sources of uncertainty. And if we look at our final formula, the traditional formula, there's no mention of uncertainty here. And so this was my motivation for thinking, okay, let's do a Bayesian solution to this problem. Moving into the PyMC code, we have our import statements and just some declarations of variables. So I've just hard-coded what a billion is. We have 25 squares in total in our visible region. And we counted 95 cells in the 5 squares. Cool. So next, we're going to set some priors and the kind of data generating process. So our first prior is, what do we think the cell density is? So up here, I've set the cell concentration to be a normal. I think that's fine because we're so far out. We expect, as I have here, 2 billion cells per milliliter. We're never going to hit a negative number with our standard deviation. So I think a normal is fine. Sort of a normal with mu 2 billion and a 500 million of standard deviation. Next, in our data generating process, we took out a milliliter of slurry. So that's represented here. On average, I take out a milliliter, but there's some standard deviation there. I eyeball it. My pipette's not perfect. Whatever. So there's some error there. Also, in the shaker, in these shakers, I expect there to be 9 milliliters of water, but it's anyone's guess. It's a bit of error on that. So I put in a standard deviation of 0.05. I think that's appropriate. Finally, the after dilution of the shaker, after I add the yeast and mix it up, I should have this quantity. So it's my 1 milliliter approximately divided by 10 milliliters approximately. That should be my dilution. Now I did that 3 times, so I do that twice more. I do the same kind of prior on the different shakers and the different transferring 1 milliliter transferring in the pipettes. And I end up with a final dilution factor, which is just the multiplication of each of those dilutions. So again, on average, it should be 1 over 10 times 1 over 10 times 1 over 10. But because of the uncertainty and the kind of measurement errors, it's going to be there's going to be some error there. And interestingly, it's multiplied. That error is multiplied. And for the hemocytometer chamber, I'm modeling it as a gamma. It should be, again, on average, 0.0001 milliliters, or I guess cubic centimeters. And there's some standard deviation on there. I think I got that standard deviation from the manufacturer, and then I think I amplified it a little bit just in case. But that's our volume of chamber. Okay, so let's talk about the next step. So imagine this is our final dilution vial, which looks like this. I have how many yeast in here? I have approximately my initial yeast concentration times the final dilution factor times the shaker volume. So it's 2 billion times times 1 over 1,000 times 10 milliliters. That should be the approximate number of yeast in here. It's a big number still because yeast concentration is so high. It's a big number. I take my pipette, I remove a small fraction of it, and I go to my hemocytometer and add it. And so what I want to know is, what's the probability that a yeast makes it from the vial to the hemocytometer? And if you just think about it, it's going to be the fraction of volume on the hemocytometer divided by the volume of the vial. So in math, again, this is the probability that I took a yeast from here and it landed on my hemocytometer, the little glass plate. Great. We also have this fact that we have millions of yeast in this final concentration, in this final vial. We have the probability of being removed is very, 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 very small, mostly because the hemocytometer volume is so small. So we couldn't model this as a binomial, the chance or the number of cells that land on the hemocytometer. But because we have these 2 facts, we can use the Poisson approximation, which states that you can approximate a binomial if these 2 inequalities hold true with a Poisson. Okay, so going back to PyMC code, we have the number of yeast visible, the number of yeast on my plate that I can see is going to look like this, and the mu kind of works out to be this quantity. Great. Now there's still one more piece of uncertainty, and that's if I have this hemocytometer, it's divided into, in this case I have 9 squares, but in my actual case I had 25. I'm not counting all of them. I'm only counting a subset. I'm counting these 5 squares. And again, we could get unlucky and all the yeast cells land on the non-counting region, or vice versa, we could get really lucky and all the, I guess it's also unlucky, all the yeast cells land in the counting region. So to model that, we just use a binomial in that case where we have the n, the total number of possibilities or coin flips, that's equal to yeast visible, the number we just calculated, and the probability of being seen is the squares counted divided by total squares. So that there's numbers up here. So in this case, if one of our yeast comes from, just make it clear, one of our yeast comes from here, lands in the hemocytometer, it has a 5 over 25, a 20% chance of us actually counting it at that point. And this in PIMC is a observed variable. We counted 95. So that's it. So we've gone from all these layers of uncertainty, and some are multiplied together, and we've landed on a single data point, 95. Now that's the data generating process. The beautiful part about Bayesian methods is that we go backwards. So we take that, this is cool. It's a single data point, 95, that's the only data point I have. And I go backwards and I look at, okay, what's the estimate of all my other variables? So with that, let's look at the posterior now. I've done 5,000 samples and we get something like this. So recall that the traditional formula gave us 4.5 billion cells per milliliter. This on average is telling us this about 4.3. And I think that's because the influence of the prior kind of pulled it towards 0 versus if we chose a different prior on the original cells per milliliter. But what's interesting and what I think is really fascinating is look at the uncertainty on this. It's almost a billion. It's huge. Our traditional formula just said it's 4.5, go run as fast as you can with 4.5. This posterior is telling us, hold on, there's a lot of uncertainty here. It could be as low as 3.5, as even as high as 5.2. That can make a big difference. And this is really important, maybe not for pitching a homebrew or even at larger scale, but it makes a big difference if you're talking about red blood cell counts or sperm counts, or if you're working in fermentation and biotechnology, like the kind of really high stake stuff. A billion just isn't going to cut it. There's other methods that they probably use or they do use to do cell counting, but this is, I think, still really interesting. So you might be saying, especially if you work in biology or biotech, you might be saying, you know, like no one uses these plastic pipettes, these things. These are kind of disposable. They're one-time use. They're more for just approximations, anyways. In a lab, you might use something more advanced like this. This is a precision micropipetter, and this can accurately get microliter levels of volume. So you'd use this to do your pipetting and your volume for your shakers, and that's true. But you're not reducing the uncertainty. You're just moving it around, and this is an analogy I come back to a lot, where uncertainty is like sand under a rug or under a carpet. You can't remove that sand. You can only push it around, and you can make it into a peak, a tighter and tighter peak, but the volume is always the original sand volume, or in our case, it's 1. You can't remove that sand. You can only move it around. So even if you're using a precision micropipetter and you have terrific volumes, you're not losing uncertainty. You're just kind of moving it somewhere else. You're making it tighter around some area, but the uncertainty is still there. So I think that's the moral of this story is that even with one data point, we can do pretty well. If we had more data points, we'd just be moving that uncertainty around. With better instruments, again, we'd just be moving that uncertainty around. But I hope this kind of this talk was interesting to you, And I thought it was a cute little toy example of a more interesting and less trivial Bayesian problem that I encountered and wanted to solve. And I hope if you do homebrew, you might actually want to use this model. Yeah, that's it. Enjoy the rest of the conference and have a great week. I hope everyone is happy and healthy out there and enjoying what we can of life. Okay, bye everyone.